Thank you. Yes, so today I'll be presenting work that I did as part of my PhD at Rice University in the Trangen Lab. The first thing I want to address is, is it progressing? The most frequently asked question I get about the software is actually about the name. People don't know what arraya is, so I will inform you. Arraya it's a relative to an ostrich or an emu, but it's from South America, and I'm told it's supposed to be a little more docile. But for our purposes here, it's an acronym for recognizing heterogeneity and evolution in assembly graphs. And so what I pictured, probably the most common use case for a tool like this would be this kind of scenario where you have maybe a human who was healthy and then they're starting to show an onset of disease. And so what we'd want to look at is their microbiome both before and after the disease. And we'll look for those subtle differences, that subtle change between our two different time points. So I was picturing probably more of a longitudinal type analysis here. Although what this tool was originally developed for wasn't quite longitudinal, it was actually it was an environmental sample of hot spring microbial mats. Within the microbial mat, the microbial mat survives at different temperatures, and there's different bacteria surviving at different temperatures. And so we were looking to try to find specific genes that seem to be driving the ability for these microbes to survive at such high temperature. And so since we were working with a not very well-studied environment, we really wanted to try to avoid using references in our software. So this brought up our tool Rhea, and what we realized as we were kind of coming up with this plan about what we wanted to do is what we were really looking for structural variants here. So I'll define what I mean by structural variants. A structural variant is a long stretch of the genome that is different between the one you're studying and some reference. So it's different from like a single nucleotide variant. We're looking for a longer stretch, typically defined 50 base pairs or more. On the left here, I'm showing a few examples of different types of structural variants. We tend to hear structural variants a lot in human genomics, looking at maybe a healthy human cell compared to a cancer cell or some type of problem like that was where I tend to see it. And in this question, what you're looking for, I think, is pretty clear. You have a defined reference, you have a defined genome that you're studying, and you're looking for these long differences between the two. And when you do this for microbial genomics, the question is still quite similar. Maybe you're working on coronavirus and we have our reference strain, and now you're studying a new population and you want to know if it's the same strain or maybe something a little different. But where I think the terminology gets a little confusing here is you have these structural variants, but now is it a structural variant of a strain we already knew about? Maybe now it's a new strain that you've discovered. So it starts to get a little fuzzy, but in isolate genomics, I think the question is still quite clear, where you have a defined reference and a defined defined microbe that you are studying. When we go to microbiome, it starts to get a lot more fuzzy. Now you have a whole community of microbes as your reference, and a whole community of microbes that you want to know. And it's hard to know which reads came from which microbe, which reads you should compare with which reference genomes, and so on and so forth. So this is what we wanted to get into, was detecting structural variants when you're looking at the entire microbial community. And so for this, our solution is to use co-assembly graphs. We're kind of cut off on the top there, sorry. So I'll define what I mean by co-assembly graph. So here we're using an assembly graph, and I've heard many different names for this type of assembly graph, maybe a quantigraph, maybe a string assembly graph. But the idea is that you have a node that is a sequence, and then if you have an edge between your two nodes, that means based on the overlap of your reads of your community, you believe that it came from a sequence that goes straight from one node to the other. So straight from CAA here to GTC, or perhaps the other direction. This is bidirectional. And there's been previous work in this space. This is a paper from 2019 that came out of Mihai Pop's lab. And they're linking specific motifs of the graph. So certain kind of substructures that you see within your entire assembly graph to specific biological variants. So we leveraged this idea to come up with ours, and I'll walk through this example of a prophage insertion, since this is a type of structural variant that I think you tend to see in a microbial community. So a prophage is or a bacteriophage is essentially a virus that attacks bacteria. And so how this works is the virus, or the phage, will attack the bacteria, and it will insert genetic material into the bacterial cell. if it goes as the prophage or as the bacteriophage plans, this genetic material will be uptaken into the bacterial chromosome. And now when the bacteria replicates, this is replicating as well. It's essentially just part of the bacterial chromosome. So if you were to build a co-assembly graph here, and what I mean by co-assembly graph is building assembly graph with multiple samples. So you took all your samples, both your time point 0 and your time point 1 samples, take all the reads and you build one assembly graph with reads from both. So what we'd expect to see here in this structure is something like this, where you have from time point 1, you went straight from bacteria to bacteria, so you'd have the top line there. But time point 0, but then at time point 1, now that you've inserted your prophage sequence, you'd expect to take the lower path where you went from your bacteria genome into the prophage and back to bacteria. And so then if we were to look at the coverage of this, if we were to align the reads from time point 1 back to this graph, we'd expect to see something like this, where you have quite a bit of coverage on your bacteria parts of the nodes that came from the bacteria genome, and lower coverage for the prophage. Since this bacteriophage has only infected one of the bacteria in your entire community, which will probably have other bacteria that are of the same species but do not have the same attack from a phage. However, if the prophage seems to give the bacteria some sort of evolutionary advantage or survival advantage within its community, we expect this bacteria to replicate and be able to survive over the other versions of it that did not have this insertion. So we'd start to see the coverage. If we were to align the reads from time point 1 back to this graph, we expect to see the coverage start to increase for the prophage, but stay relatively consistent for the bacteria. And so if we look at the log-fold change, or you want to look at the change between time point 0 and time point 1, for this, we've chosen to use log-fold change. You start to see this pattern. So essentially what we see here is a triangle with one outlier, with one point that has a significantly different log-fold change in the coverage between the two time points relative to the other two. So we're looking for one outlier within this triangle substructure in the graph. So this brought us it's kind of the underlying idea of our entire array of pipelines. So here's the whole pipeline. We take our samples here. I'm using time 0 and time 1 as essentially like a longitudinal, although as I've alluded to in the previous slides, it doesn't need to be longitudinal. You just want your communities to be very, very similar. You have some reason to believe that they're quite similar. We build a single co-assembly graph. Since we were using Oxford Nanopore data, a little bit older Oxford Nanopore data, so it's somewhat relatively high error to what Nanopore seems to produce now. So we use Metafly to get a single co-assembly graph, and this is just using like default Metafly, but putting in all your samples into one assembly graph. We then align each of the set of reads to the graph independently. So this way you can go back and look at your coverage for the graph, your coverage for each node for each specific time point, here being 2 different time points. Then, since the graph is the same, you can look at the change in coverage over the node between the 2 different time points. And then we can go into this graph and look for specific patterns. So I'll zoom in here briefly on the types of patterns that we look for. Within Rhea so far, we have 4 different structural variants that we detect. The first 2, insertions and deletions, we look for a triangle where one node is an outlier. So for an insertion of you, one node that has significantly increased relative to the other 2 points in the triangle, this is where we'd call an insertion. A deletion would be the opposite, one node that has a significant decrease. So what that means is over time, for some reason, the sequence or the genes in this node seem to be increasing or decreasing relative to its community. The third one here, CI here, stands for complex indel, and what that means is you have an insertion and a deletion at the same location. So for some reason, one path is being chosen over the other one for survival. Then lastly here, TD is for tandem duplication. So this is a direct repeat, one right after another. And we detect that by looking at a self loop in the graph that is increasing in its coverage. I get a lot of questions here about the runtime and if this is feasible. So this is, we've developed it with Python. So we use NetworkX and we use simple cycles to detect all the triangles and squares within the graph. The biggest factor that's going to slow this down is the average node degree. So if you have a graph that is very complex, and by complex I mean a lot of edges, that's what's going to slow it down the most. And so in the sequences I've at least worked with, for the hot springs, it was a really complex community. So that was taking probably like a couple days. It was running, but it was quite slow. For the communities that I think are probably more what people would use, the ones I was working with was a cheese microbiome. This was pretty quick, just taking maybe an hour or two. Okay, into a couple simulated test results. This is a Zymobiomix even distribution. So I'm sure many people in here know and love the Zymobiomix mock community. It has 10 different microbes in it, 8 bacteria and 2 fungi. And even distribution, it's a relatively even distribution. It's even for the bacteria and then a little less for the 2 fungi. And so what we did is we simulated a variant for each one of our 10 microbes. The variant introduced 40 different structural variants, 10 of each of our 4 different types. And then for our time 0 time point, we used no variants, only the original references that come in the Zymobiomix mock community. For time 1, for half of them, we introduced only the structural variant, so you do not have the original in the community. And for half, we've introduced both an even split of the variant and the original. I think this is more representative of what you'd expect to see in a microbial community. We tend to have many species that are or many strains even that can be quite similar. So we do want to make sure that our algorithms can handle that. And so as we expected, once we've introduced multiple variants of similar strains, that's where it starts confusing in existing algorithms. So as far as I was aware, there's not really a lot of work in this space. There's not really a lot of structural variant detection methods that are for Mihai saying maybe none at all based on the nod I'm seeing. And so what we did in place of that was use what I've seen in a few papers where they essentially they make mags from their communities and then use a single structural variant detection method that is used for a single genome between the mags. So that's what we've done here for our comparison. So on these graphs here, the yellow or the orange bars show what we got out of our mag workflow, and the blue here for our tool with Rhea. And so the first thing you notice is that the mag workflow only generated mags for 5 out of the 10 microbes in the community. So now you've already really limited your analysis to be only those few set of microbes. But in the situation where the mag is generated and only the variant was present in time 1, I did find Rhea and the mag workflow to be relatively similar in their performance. This graph here that shows the recall for only the specific species, it does show Rhea outperform mags, but I think that's mostly because Rhea was able to detect complex indels and this mag workflow wasn't really able to, so that's kind of an unfair bias there. But so I think they're quite similar. However, what Rhea really seems to outperform the mag workflows is when multiple similar strains are in the community, which I think is the big problem and difficult thing when working with microbiome communities. We also did a log distribution. This was a similar test, but for a very different reason. This was to see at what point we start to see how much coverage you need for Rhea to be able to detect the structural variant, essentially. And we found that with 4x coverage, which was in this Bacillus subtilis species, Rhea was still able to detect about a quarter of the structural variants. However, once the coverage decreased much lower than that to 0.4x, we're not able to detect any of the structural variants. All right, so to recap here, Rhea is a software algorithm for structural variant detection and exploration via long-read co-assembly graphs. I didn't really talk about the exploration portion, but essentially the output of Rhea will allow you to go into Bandage and you'll get a visual. That way you can see more of the context of the structural variants that are being called, and perhaps maybe you want to do some other exploration. You'll be able to do that with the output of Rhea. The benefits and things that I like about this tool is that it is reference-free, which is really nice when you're working with newer communities. The fact that we're using a co-assembly graph here is a high inclusion of reads as opposed to using something more downstream that starts to discard many of your reads. And this allows us to handle more diverse and complex microbes. And some limitations here is right now we're only detecting 4 different types of structural variants. I do think this setup, it does have room to grow. It does have space to expand to other types of structural variants with a little bit of thought of the types of patterns that we would expect those structural variants to produce in the co-assembly graphs. It also requires series data with similar long read communities. So far, we've only tested on long reads. I think if this were to expand to short reads, it would take a little bit of thought as we'd expect the graph to be much more complex. And also, I've said this, that the communities need to be quite similar, and we don't really have a clear definition of what we mean by similar. And so maybe it's hard to know exactly like where this should work and where this will start to fall off. But if you're to take away anything from this presentation, this is actually what I would hope it would be. So here you have a microbiome community. It's rife in different microbes that are interacting, that are influencing each other, and it is growing and it is changing as a community. But so often in microbiome analysis, I think we try to make it easier computationally, especially as computer people. And so we tend to break it apart, and we tend to look at only one microbe at a time. But truly, I think this quote applies to microbial communities, that the whole is greater than the sum of its parts. And so what I encourage you is that if you're working with microbiomes, to remember that you are working with a community-oriented system that is more than just a sum of its different parts. So I would encourage you to try to use software that view the community in that light. With that, a huge thank you to my PhD advisor, Dr. Todd Tringen from Rice University, and our lab. Also, a thank you to the many collaborators we had on this project from Institut Pasteur, from Rice University, and from Carnegie Science. And thank you to ISMB for providing me with the travel award that has allowed me to make the trek from France here today. Thank you. I'm happy to take any of your questions.
For questions, please come up to the microphones.
Thank you. Yeah, brilliant talk. I was wondering, it's not my field of expertise, but is there any possibility for larger graphs that you can improve runtime practically if you prune it? Like, are there edges that you can remove that you deem to be not as important as others?
Okay, so the question is, can we improve the runtime by decreasing the graph, essentially, by pruning the graph? Probably, but I just think a big advantage of a method like this is the inclusion of reads, and so I just feel like you'd be throwing away potentially your signal. So I guess if you're really tight on time or space or whatever computational requirement is getting to you, you could probably find something like that. I'm not convinced I'd want to do that. I think it would be, I don't know. The point is to try to include as much as you can, I think. So, yeah.
Thank you.
Thanks, Leo.
And one more question.
I also had a question that kind of maybe goes against the purpose of it, because cool. I was thinking like, well, could you do, even if you didn't like split things up by a reference very strictly, could you maybe like gain some speed advantages by doing like a very like high level phylogenetic classification of some of the reads? Like, you don't have to know what species, but maybe like if something really looks like, especially because you have long reads, if something really looked like it was in a different phylum, maybe you would be able to skip trying to assemble. I don't know.
Yeah. So I think the question is the kind of pre-filtering or pre-separation, so that way you're doing the assembly on only a smaller portion of the reads. Cool. I think that wouldn't make a big difference in practice, because I think your assembly graph is going to put those reads into different connected components anyway, and where the time, where we're slow is the nodes that connect to a lot. So I think they would, hopefully the assembly would kind of separate those out anyway.
Got it. Thank you.
Yeah, thank you.
Let's thank